అది పంచాయతీ చెప్తావా? ఆ ఇంటి వచ్చేసింది జ్యోతి
Huh?
లక్ష్మీదేవి చెప్పిండు లావుతున్నాడు.
Yeah, sharing is fine.
Sharing is
నేనే చెప్పింది. మేడం
Thanks, Mari.
గుడ్ మార్నింగ్ ధనంజయ రెడ్డి గారు.
Sir, good morning, sir.
Good morning, sir.
Good morning, madam. Good morning, all of you.
Sir.
మార్నింగ్.
Good morning, sir. D.
Sir, good morning, sir.
Good morning, Chalpati.
Yeah, good morning.
Sir, 2024 ki
Sir, good
24
morning, sir.
last meeting ఏమో sir. 2024 లో లాస్ట్ మీటింగ్ ఏమో sir.
Last meeting is it? Oh, no.
Sir.
ధనంజయ రెడ్డి గారు,
Start చేద్దాం.
వీసీ గారు తర్వాత జాయిన్ అవుతా అన్నారు, మనల్ని కంటిన్యూ చేద్దాం.
సరే మరి స్టార్ట్ చేద్దాం సార్.
స్టార్ట్ చేస్తాం సారు, తర్వాత జాయిన్ అవుతాం.
అలాగే సార్. ఓకే, థ్యాంక్ యూ. Good morning, all of you. Welcome to Dr. YSR Horticulture University, Venkatramanagudem, West Godavari district, to the 20th Global Connect series in collaboration with West Virginia State University, USA, on genetic basis of anthracnose resistance in tomato. This is the topic. And today, our guest speaker, Dr. K. Umesh Reddy, Professor of Biology, West Virginia State University, USA. Good morning, sir. Welcome to Dr. YSR Horticulture University. So, good morning and welcome to our Honorable Vice Chancellor and Chairman, today's Global Connect Series Chairman, Dr. K. Gopal Garu. And with his visionary and dynamic leadership, we have arranged the 20th Global Connect Series today. And thank you, sir, for giving permission to conduct a Global Connect Series. Sir is very dynamic and very positive. And sir wanted to have a linkage with the international community. We are planning to have MOUs with almost 20 international universities and under his leadership. And we are linking with industry and international with the Horticulture University. After joining in charge, sir reviewed this DIP section and we are going in big way. So as a result, recently we have signed a MOU with the Mondeleza Cadbury International. We got 13 lakhs rupees of scholarships to the BSc, MSc and PhD students under visionary leadership of our Honorable Vice Chancellor, Dr. K. Gopal Garu. Thank you, sir. Thank you for giving this opportunity. And today guest speaker, Dr. K. Umesh Reddy Garu, Professor of Biology, West Virginia State University. Thank you, sir, for accepting this our invitation. You are the guest speaker, first guest speaker 2 years back. And with starting from you, so we are successfully conducting 20th Global Connect Series, sir. Thank you very much for accepting our invitation. And welcome to our all university officers, Dean of Horticulture, Dr. Narman Naidu Garu, Director of Research, Dr. Madhavi Garu, Director of Extension, Dr. Govindarajan Garu, Registrar, Dr. B. Srinivasulu Garu, Director of Planning and Monitoring Cell, Dr. Vijaya Padma Garu, Dean of Student Affairs, Dr. Suri Kumar Garu, Dean of PG Studies, Dr. K.T. Venkatramana Garu, Controller of Exams, Dr. Prasanna Kumar Garu, and University Librarian, Dr. Ramanandam Garu, Controller, Dr. Srinivas Subramaniam Garu, Estate Officer, Ramana Garu, all Associate Deans of Colleges of Horticulture and affiliated and constituent colleges, Principals of Polytechnic Colleges, and Zonal Research Heads of Coastal Zone 1, 2 and Rayalaseema, and all heads of agriculture research stations and KVKs. And today, rapporteurs, Dr. C.H. Nalini, Assistant Professor, Entomology, College of Horticulture, Venkatramanagudem, Dr. S.B. Ramya Lakshmi, Assistant Professor, Agriculture Economics, College of Horticulture, Anantarajpeta, and vote of thanks by our Dr. B. Srinivasulu Garu, Registrar. And before going to details, just we will have a brief introduction of our guest speaker today. And Dr. K. Umesh Reddy garu is a distinguished professor and a researcher in genetics and plant breeding, biotechnology, and genetics at West Virginia State University, with a robust academic foundation, including a PhD in genetics from Osmania University. He has made groundbreaking contributions to crop improvement and genomic research. Dr. Reddy has held esteemed positions, including an assistant research scientist at Texas A&M University, where he developed genome-wide markers, constructed genetic maps, and advanced marker-assisted selection for breeders, significantly influencing the germplasm enhancement and molecular biology research. Dr. Reddy has secured over $18 million in research funding and published over 150 peer-reviewed papers in high-impact journals. He leads a vibrant research team, supervising 8 postdoctoral associates and 10 graduate students on innovative thesis projects. An advocate for global collaborations, he has hosted scientists and students worldwide, fostering an inclusive research environment that bridges cultural and academic boundaries. A recipient of numerous accolades, including the Distinguished Scholarship Award, Scholar awarded at West Virginia State University 21-22, Professor of Year 18-19, and Rockefeller Foundation Fellowship. Dr. Reddy exemplifies excellence in research, mentorship, and leadership in agriculture genomics. Thank you, sir. Welcome to Dr. YSR Horticulture University 20 Global Connect Series. Sir, wife also, the personal details are born in Pulivendula and Kadapa district, and wife Dr. Padmavati is a professor of genetics and plant breeding at the same university, West Virginia State University. Sir having one son working in US intelligence services. And thank you, sir. Thank you very much. And now our Honorable Vice Chancellor will join shortly, Dr. K. Gopal garu. Meanwhile, you can proceed, sir. Thank you very much. Hello?
Yeah, starting.
Okay.
So thank you, Dr. Rao and other dignitaries for giving me this opportunity, and also glad to know that as some professor suggested that this is the last meeting of the university.
For this year.
This is
For this year.
Yes. So can you all hear me? My voice is coming.
Voice okay.
తెలుగు సార్ ప్లీజ్ సర్వీస్ వరకు అవుతుంది మాకి ఎజనల్ చేంజ్ అంటే టెక్నీషియన్ భువనేశ్వర్ నుంచి చూస్తాను బండి తిరిగింది అని చూస్తాను
10,000 తిరిగింది
10,000
ఫ్రీ సర్వీస్ ఉంటుంది థర్డ్ ఫ్రీ సర్వీస్
చూస్తాను.
Can you all hear me?
Yes, sir. Yes, sir. Proceed.
Okay, so however, my topic today is genetic analysis of anthracnose resistance in tomato. We all know what is anthracnose. You are horticulture professors and scientists. Probably you are dealing this in several vegetables and fruits. So in tomato is the most popular vegetable, after potatoes. In 2022, it's almost 1.16 billion US dollars. And then the anthracnose is caused by Coletotrichum species, a major disease for the Solanaceae family, for the peppers, tomatoes, and even many other fruits and vegetables. So mostly, this can be managed using pathogen-free seeds, crop rotations, and well-drained soil, and any alternative hosts are there, such as weeds that can be controlled. So the anthracnose is an ascomycetes and it produces conidiaspores, ascospores. Conidiaspores are the asexual spores. Ascospores are sexual spores that are result of reproduction between plus and minus mating types, and then the anthracnose is affected when the fruit is green,
ఇవి ఉన్నాయి కదా అర్జెంట్ ఇది లేకపోతే ఏమయ్యేదంటే
and then this biotropic colonization of then it will be quiescent, okay, till it ripens. Once it is ripened, it will show up. Also, it can form the bodies like Eservulae. So the Eservulae also produces conidiaspores, and then it can spread the disease. So probably you have very important seasoned pathologists. They know much more than what I know about the life cycle of anthracnose. So in short, we can control anthracnose or build resistance through management or improvement. And then understanding, the plant pathogen interactions is important for developing resistant tomato varieties for anthracnose. So the research objectives for this project is to identify genomic regions for anthracnose resistance using techniques like QTL mapping and then QTL-seq, and then validating those results using these PACE assays that can be used for high throughput breeding or germplasm characterization. And then once we have the candidate genes, potential genes, these underlying mechanisms, how they regulate anthracnose resistance, can be understood through creating knockouts using CRISPR technology. So this project is started with a very susceptible US-28, a very famous cultivar, tomato cultivar, which is susceptible and highly resistant variety, which is Lycopersicum and Serrasiforme, probably 95L368 is a resistant line. They crossed and a rill population is developed. So what is rill population? It's a recombinant inbred line population. So in F2, taking some random plants and then selfing them and taking just one single seed per plant, and then forwarding it to several generations. So at F7 or F8 generation, we have 243 rills. So that kind of rills development is tedious, but that helps to map genes much more precisely than using F2 populations, because most of the heterozygosity is almost fixed in producing these rills. And such rills, DNS was isolated. DNS were they were used to make a library by digesting a set of enzymes, and using that enzyme-specific adapters, we can do like PCRs that important for the next-gen sequencing. Genotyping by sequencing is a trivial technique. Probably many of your genetic students knows how to do this genotyping by sequencing, which is reduced representation of genome and getting some SNPs that are segregating across these 243 lines. So using this GBS, we have developed about 5,000 markers, 5,000 SNPs, and that are used for the QTL mapping as well as GWAS. Both softwares, both pipelines were explored for identifying the QTLs, and then 30 extreme resistant and 30 extreme susceptible of this rill population were bulked. You see, once you have this DNS, you can quantify equimolar concentrations of DNA and can be bulked. So 30 of them are resistant individuals bulked as resistant bulk, and 30 of the susceptible individuals, that is having lesion size much larger. Susceptibility is measured with the lesion size. If the lesion is larger, They are the susceptible types. They are mixed. And then the analysis was performed that is called QTL-seq analysis. And then we will discuss why we did QTL-seq analysis once we did this QTL analysis. And then from this QTL-seq analysis, important SNPs were converted into assays, PACE assays. These PACE assays can be used for high throughput screening. Large populations, segregating populations, breeding populations can be screened. And then once we have identified some of these genes were validated. We are in process of validating using this CRISPR-Cas9 technology that uses tissue culture, like Agrobacterium co-cultivation, callus production, Agrobacterium co-cultivation, and then transformation and then callus induction, shoot production. So the typical tissue culture pathway will be used for developing the CRISPR lines. So the RILs, once you have this highly resistant parent, highly susceptible parent, you think of this is the black line is the susceptible genome, and totally this white is the resistant genome. And so in the F2 population, there will be segregation. So these chromosomes are shuffled here. And then one single seed from each of this plant is forwarded for the 243 plants till F7. And then we did the GBS. And then this Coletotrichum coccidis is the pathogen that we have used. A droplet is used, Coccidis inoculum, having this many conidia per mil on the fruit by puncturing through the droplet with a pipette to a depth of approximately 2 millimeters. After 6 days of inoculation, fruit lesion diameter was measured. So, and adjusted it to nearest 0.5 millimeter. So the CRISPR, as I said, we will discuss more about CRISPR, how the Agrobacterium transformation was done, how they were kind of kept for the co-cultivation, and then used for the regeneration media and produced the plants, and having the T0 plants and the T1 plants. So 3 different genes were explored. These genes were identified using the mapping techniques and QTL-seq. So we have all these 7 plants, 5 plants, 2 plants, transgenic plants that we are exploring further. So this lesion size, you see in the susceptible plant, it is very large lesion. Comparatively in the resistant plant, that is very small lesion. So the 2 seasons that were kind of screened, 2 seasons, both seasons, they were inoculated. So the resistant plant has 0 lesion, almost very small lesion, whereas the susceptible plant has almost 17 or 18 millimeter size of lesion, very large lesion because it is susceptible. So both seasons it was exactly same. And then the segregation was noted, and you are seeing here frequency distribution. So once we have all these GBS markers, SNPs that are identified using genotyping by sequencing, there were 5,000 markers, 5,000 SNPs that were used to build a linkage map. These are the linkage genetic map. So tomato has 12 different chromosomes. So each chromosome specific markers are coalesced into a linkage map. And these numbers are the chromosome that position, nucleotide position. So one thing I want to clarify here, though there are 5,000 markers, the markers that are cluttered at one place were eliminated. So these markers are called framework markers. So what is a framework map? So at framework map, at every regular interval, there is a map, there is a marker. So if this is the road, these are the signposts. For example, from Tadepalligudem to Vijayawada, whatever the towns that come will have some names for that, right? For each village or each small town will have a name. So similarly, all these genetic markers, once they are mapped, they are signposts on these linkage maps. So all these different linkage groups are representative of various chromosomes. And then we used QTL analysis So QTL analysis is nothing but for each marker, there will be two different kinds of alleles. One allele coming from resistant parent and one allele coming from susceptible parent. So if you take mean of that resistant parent in the RIL population and mean of the susceptible parent for the susceptible alleles in the RILs, if that is kind of associated with the trait, if it is linked, then it will show up that there is a QTL in that place. Or in other words, for every marker, there is a probability of being linked or not linked. For example, this marker, what do you ask it? Whether it is associated or linked to the disease or not linked to the disease. So the probability of being linked and the probability of not being linked, that ratio is called LOD, logarithm of odds, LOD. You see, for each marker, there can be two probabilities. Whether, for example, what would be the story of this marker? Whether it is linked or not. If it is linked, if it is not linked. So the probability of being linked to the ratio of probability of not linked is the LOD. So these markers, this red and green, are two different years. So some of them are showing both years and some of them are showing single year only. So you see here so many genomic regions, so many places you have the markers that are linked with the disease resistance. Okay, so what would it tell us? Anthracnose is not represented by a single gene, that the multiple genes from all across the genome are collectively contributing to the resistance. So this kind of multiple genes affecting one single trait are quantitative traits. And the loci that are contributing to those traits are called quantitative trait loci or QTLs. So QTLs also can be seen here. So especially wherever the regions chromosome 2, chromosome 6, chromosome 10, chromosome 9, all these things can be seen as a peak here. Here, what are you seeing here? Just now I told you what is LOD, the probability of being linked and the probability of not being linked. If that ratio is more than 3, there is a cutoff here. If it is more than 3, that means it is highly associated, highly linked. So chromosome 11 has a QTL position. Chromosome 6 has some QTLs. Chromosome 10 has a lot of QTLs that are blurred. And then you are seeing these peaks. So under the peak, there are SNPs that are linked. So the SNPs are located into the genes. So we know there are some genes here. So you know how many genes could be there just in this region, say chromosome 6, under this peak. How many genes can be there? Hundreds of genes can be there. So how do we know which gene is really contributing to anthracnose? Here, for example, you are seeing a peak here. There may be top peak SNP here. 5,000 SNPs are nothing in the genome like tomato. So under this peak, there can be hundreds of genes. Okay, so we need to figure out which is the actual gene that is contributing to the resistance that gave this peak. So you remember what are these peaks? These are the peaks, LOD peaks. What is LOD? Logarithm of odds. What is logarithm of odd? The probability of a marker to be linked and the ratio of probability of that marker not being linked. If that ratio is more than 3, that means the LOD is this one, having very decent LOD. So we can still understand what is happening here. So we identified there are several QTLs here. These are all contributing to the resistance. Chromosome 2, chromosome 3, chromosome 6, chromosome 8, chromosome 9, 10, 11, so forth. And then this is the interval, peak interval in centimorgans. Okay, and this is the LOD. So having the higher LOD, say 21.11, is a very decent LOD, very high probability LOD. And 25.91 is very high LOD as well. And some of them are just barely significant. So once we know these markers, whether these alleles are negatively contributing or positively contributing. So once they are additive, that means they are all contributing to each other. Additive traits in the quantitative traits, they add up. So when there are several genes that are spread across the genome, additive inheritance can be noted when we are adding up all those genes. So having the negative QTL means what? The lesion size is large. So having negative is better for the resistance in the sense if the lesion size is large, then it is showing higher LAD, higher additivity. So negative additivities in the sense they are related to smaller size of the lesion. If you understand that, if the larger size of if the marker is linked to the larger size of the lesion, then it is a susceptibility marker. But if you are seeing it like fruit weight, additive traits negative means then that fruit weight is lower fruit weight, but resistance is having the least lesion size is better for resistance. That's why additivity should be towards negative side. And then the marker is located between these nucleotides on these chromosomes. And this is the most important here is the phenotypic, variance that is explained by the marker. So some of the markers are explaining 11.9% resistance. This is 17.2%. Okay. And then on chromosome 6, this is 9.1, 6.7, 6.9. So these are the percentages. So you add up all these percentages, roughly it is 40 to 50% of the resistant markers. You know that where they are located at least, but still you do not know where are those genes. But you know that under these peaks, there are hundreds of genes, and you know the markers, there are markers located into these genes that are tightly linked with the resistance. So the same analysis can be explored using the GWAS. And here is the peak. For example, chromosome 1, 2, 3, 4, 5, 6. 6 has the largest peak. So the earlier analysis deals with the linkage, and this one with the association. Okay. For example, we can say Chinese people are yellow color. White people, Caucasians are white people. South Indians are dark people. North Indians are brown people. So this kind of associations can be validated. That association statistics is a simple linear linear model. Okay. So this one is the strength of association, having the larger 12 in the sense this chromosome 6 markers are highly strongly associated. So the previous analysis also had chromosome 6 had very strong associations. So we understood there are very strong associations on the chromosome 6. So we can understand the allelic effect. So having at this nucleotide on the chromosome 6, this allele can contribute a mean of 5.5 lesion size, 5.5 millimeters. Whereas having CC at the same position, cytosine at the DNA strand, that can have at least 10 the lesion size. So the lesion diameter you can see here. Here it is, CC is the resistant allele, and here is the susceptible allele having the larger lesion size. And then at this SNP, there is a wide difference. The difference is not much. Here it is 5, and probably this is about 8. And whereas this marker is 10, and the resistant allele is having 5. So you have these resistant alleles. So cumulatively, if all these alleles are present, that cultivar will show the resistance. That segregant will show more resistant comparatively to the cultivar segregant or the cultivar that have more susceptible alleles. So once we have these associated alleles, still we do not know what are the genes underlying this. Okay, so for understanding what are the genes that are located under these peaks, we performed this QTL-seq analysis, where 30 individuals of resistance, highly resistant, and 30 individuals are highly susceptible. Okay, in the season 1 and season 2. So here you see the resistant alleles having 5 millimeter size, all of them are here, and susceptible alleles are here, whereas the total population is kind of spread across. Okay, so this bulk specific DNAs, equimolar concentrations of DNAs are mixed and then deeply sequenced, almost 500x sequence. So here these bulks, the lowest bulk that is resistant, highest bulk that is susceptible, are mapped to the genome. So now we can understand for at a given SNP. Here, for example, this SNP is located all over. Everything is G. Okay, that is the resistant allele. So sorry, susceptible allele. Because of this G, this particular locus is very susceptible. Let us suppose like that. And then there is a mutation, and here is the C, that is resistant SNP. So, but the resistant SNP, this is coming from the bulk. So still some of the resistant individuals will have G and some will have C. So for all the loci, if there are about 100 loci contributing for anthracnose, you cannot expect at each and every loci, resistant individuals will have totally homozygous. So a individual will have resistant loci here. For some other SNP locus, it may be even susceptible. Okay, that's why they are called quantitative trait locus. Now SNP index can be calculated. For example, there are four different SNPs that are in the resistant parent, that is divided by 10, that are 10 in the susceptible individuals, because these are the sequences we got for this particular locus. These sequence counts are called BAM counts. Sometimes 300 BAM counts. So 300 BAM counts means 300 different sequences that we got from the bulk. So using this kind of SNP index, you can calculate delta SNP index. Delta SNP index is once it is mapped to the parent B, or a resistant parent, or susceptible parent. So when it is mapped to the A genome, that is the susceptible parent, if it is -1, that means all these SNPs that are you have seen in the resistant bulk are exactly, all of them are C. Then it is -1, because 10 by 10 equal to 1. If it is resistant genome, it is B. When it is susceptible genome, it is A. In the sense, -1, in the sense, all the resistant SNP is replaced, this allele is replaced with the resistant SNPs, Cs. There is no G at all. That is 10 by 10. That is -1. Okay. So this is how the delta SNP index is calculated. So you can understand here, these are those SNPs. Because it is 500x genome sequencing, you can see these peaks. Here especially, I will ask you to focus on the chromosome 6, where you had biggest peak in the GWAS. So that peak is here, okay? So, and then in the susceptible genome, this peak is exactly opposite. So you are taking 10 susceptible alleles divided by 10 resistant alleles, then you have the peak is positive. But if you are taking 10 resistant, 10 susceptible, 10 resistant alleles divided by 10 susceptible, then it shows up negative. In nutshell, in summary, without confusing you guys much, having a negative SNP when you map to the susceptible parent indicates that that is a resistant loci, very highly resistant loci. So once you have identified all these resistant loci, and now you can compare the QTL mapping and the GWAS and understand what are the common regions that harbor the genes. So for the QTL-seq is very precise. It will show up the genes, genic locations. Because it is bulk sequencing, so 500x genome sequencing will give thousands of, millions of SNPs. And these millions of SNPs can be compared with these positions, and then these common positions were noted. So for example, on this chromosome 6, there are these common positions. But still, in the QTL-seq shows on the chromosome 6, there are about hundreds of genes. It's not just few genes, but you can precisely identify what are the SNPs that are showing the resistance now. So those SNPs are converted into PACE assays, that is called PCR allele computative extension. That is, I should have made a slide for this. For example, you have a resistant SNP, you have a susceptible SNP, and then you keeping that SNP at the terminal end, you will have to design the primers, and a common reverse primer can be used to amplify them. So this kind of PACE assays will discriminate. So at least you will understand this. The resistant individuals will show blue, and then susceptible individuals are shown here green, and then red, and then heterozygotes are green. So these are called allele discrimination plots. So for all the markers that are on the chromosome 6, because chromosome 6 is very important, that has a lot of important genes in it. So we need to identify how those genes are kind of being useful for further, if we want to use them as DNA markers for selecting resistant lines and susceptible lines, we can create, using this PACE assays, PCR allele computative extension, in the sense PACE. These PACE assays will create this kind of discrimination plots, allele discrimination plots, and then that kind of data can be subjected to single point analysis, which is called Kruskal-Wallis method. So if some Kruskal-Wallis shows highly significant chi-square, that means that particular locus, SNP locus we targeted, is associated, very strongly associated. For example, here you can see it here, A allele and B allele is highly discriminated, significantly discriminated. Here is the lesion diameter. So the A allele that is coming from the resistant parent is having very less lesion size comparatively to the B, which is coming from the susceptible lesion size. So without inoculating with the disease, folks, very important for geneticists and pathologists and breeders, without inoculating with anthracnose, you can identify the resistant individuals just by using these PACE assays. If the lesion size is small, that means that is coming from the resistant locus. Okay, so what are the most important genes on the chromosome 6? There are plenty of genes, few are listed here, but how do we identify which is actually the gene that is contributing to the resistance? So then we have to look for the literature. For example, a study happened, 2 different studies. They used this ethylene responsive transcription factor, an important gene that helps ripening of the fruit, over-ripening of the fruit. If that gene is overexpressed, then it can cause susceptibility. That is understandable. When ethylene responsive transcription factor upregulates, the fruit becomes ripened so fast, and then it would be susceptible only. So another one is NAT-A is another gene which we have identified in that QTL-seq as well as QTL analysis, that it regulates plant defense mechanisms through acetylation. So the genes need to undergo acetylation before transcription. So this acetylation is kind of administered by this acetyltransferase that affects pathogen resistance, and that study was done in 2023. And here is another transcription factor, which is called BHLH35, and which is also very important for infection in soybean. So by looking at various studies, this is done in pepper, this is a general molecular study, this is done in the soybean, we could understand that these 3 genes must be important for the tomato, doing the similar kind of functions. So now we started genome editing. Genome editing is, first you need to identify such traits and you can target those traits. So if you know that we can design a guide RNA for each specific gene. A guide RNA goes and targeted its homologous sequence. Here is the endogenous gene for which this guide RNA targets, and this guide RNA is fused with CRISPR RNA, which can kind of can interact with a gene called Cas9. So this is a technology. It's not a huge rocket science. By looking at few YouTube videos, probably you can understand how the CRISPR works. So CRISPR starts with a design for the first you need to target a gene. For example, if you want to target these 3 genes, you need to design the guide RNAs. And then guide RNA you need to clone using this Golden Gate technology. Here it's pretty easy cloning. And once you clone it, it enters into this vector. So that vector you can use it for transformation of the plant. And then you can select those and then identify the edited plants. How this guide RNA works is guide RNA goes and targets its endogenous gene, the gene that is present in the tomato, and can cut it. So you can remove that gene. You can silence these genes. So this is one of the revolutionary technologies that are very cheap. Anybody can do it, that do not cost much. By reading some papers, you can identify the genes and you can target this technology. And then using this technology, silence genes. So for example, these 3 different genes are nothing but DNA sequence. That's why you see this bunch of nucleotides here. A guide RNA is designed and it needs a kind of a triplet nucleotide sequence that is called, photospacer. And then that is put in a kind of construct. So the guide RNAs are fused with a U6 promoter, which is U6 promoter is very important to tag with the guide RNAs because RNA polymerase 3 is the one that transcribes, that acts with the U6 promoter to transcribe this kind of small guide RNAs. So once you want to transcribe small guide RNAs, you need to fuse this guide RNA with the U6 promoter. So 2 guide RNAs, flanking guide RNAs were sequenced. So 2 scissors, one scissor this side, one scissor this side. You can design it. And then that is in the same construct, you have cauliflower mosaic virus 35S promoter, which is regular promoter. So which acts with the RNA polymerase 2. So this Cas9 is Cas9 specific RNA is produced with this promoter, whereas U6 promoter produces this guide RNAs. So there will be 2 different transcripts. One will be a transcript coming from this promoter, which is for the Cas9. Cas9 works as a scissors, and then U6 promoter produces this kind of guide RNAs. Here it is a single guide RNA used for different transcription factors. And then the results are like this, the agrobacterium transformation, and you do the colony PCR. This is the routine things. You don't have to have very sophisticated laboratory to do this. You can do it even in the Tadepalligudem with very limited budget. So you can silence some of the bad genes. So the most important technology is just doing standardizing this kind of tissue culture and transformation, which any tissue culture scientist can able to do it. So the molecular geneticists can create this kind of constructs that is putting guide RNA fusing with the U6 and Cas9 with the 35 cauliflower mosaic virus specific promoter, which creates 2 different transcripts. So now it should be a single band here. What it shows here is this band is not cut, but here that cut has made this band smaller. Okay, but still some cells are there, uncut cells, and here is still uncut, and here is the cut. So this is the full length gene which is uncut, and this is the gene in between that gap that is cut. Okay, so using this kind of technology, we cut these genes. So once you cut a gene, that will not function the way it should. Like if I cut someone's legs, whether one can walk, cannot walk. Similarly, the genes that are deleted or removed a portion of it will become ineffective. So the AP2 genes, AP2 is the transcription factor that can make tomatoes to ripen very fast. So this kind of this is the kanamycin using which Cas9 could be identified. Yes, Cas9 is there. So Cas9 is the scissors. So how this scissors works? You need to have a guide RNA. This guide RNA1 and guide RNA2, these two are targeted with the Cas9. So wherever this guide RNA is fused with this endogenous gene, this Cas9 can cut it, chop it into fragments. Now this is little bit larger T1 plants you can see here. Is this cut? It is not cut. This is cut because it became smaller. This is not cut. These are all four are cut. You see, this is not cut. These are cut. So once you identify this kind of edited plants, you can make sure that the Cas9 is eliminated in the segregation. Okay, because if the Cas9 is there, it will be repeatedly throughout the generations, it will be cutting. So you don't want that. So you just cut once and keep that plant if you segregate out Cas9. And then BHLH, since we used one single guide RNA, it is not cutting it. So if you see the gene is like thread, if you are cutting at both places, it becomes shorter. Here, this guide RNA is not cut that way, but it has produced some kind of same size bands. But if you sequence, you will see some single nucleotide differences here are small differences. Okay. So here you can see in the BHLH, in the AP2, you have large deletions because you have used two different flanking guide RNAs. So you created 794 base pair deletions. And then here is probably it is with single guide RNA. That's why it has created small deletions. Okay, because cutting at one end. So this PAM sequence has to be there where the guide RNA is designed. So students come to our lab that just from the Pondicherry University, or we have a student from the YSR Horticulture University from Kodur campus. And they're all doing these technologies. It's not any rocket science. It's very easy to do it. So this is the paper that we published. So if you don't understand my talk completely, you can also read this paper. This is freely open source in the Frontiers in Plant Science. You can just go to the Frontiers and Plant Science and search. You can say QTL and PACE analysis for the genes, anthracnose resistant genes in tomato. You can see this paper. So what we did so far is we did QTL mapping, a technique called GWAS, and validated those results with QTL-seq, identified the major QTLs and the genes that are on the chromosome 6, and created PACE assays so that we can use them for, so that breeders can use it for selecting individuals or the breeding populations for the resistant alleles. And then we used various genome editing technologies to silence some of these genes. So the fruits will be collected. Currently, a student and a postdoc are working to collect the fruits. Once we have the fruits, we will inoculate and see what happened once we cut these genes, how that worked. So this is our lab, Dr. Padma Nimmakailas and mine joint lab. Carlos is doing PhD in this project. Saiti Ramireddy is from Pondicherry Agriculture University, who created most of these CRISPR lines under the guidance of Subramaniam Chinreddy, who is very efficient postdoc who created all these CRISPR plants. And Wagner is a distinguished tomato geneticist from West Virginia University, which is a larger campus, and who is pioneer in all these technologies. And John Stommel is USDA scientist who collaborates with us. Thank you.
Thank you, sir. Thank you very much. Excellent presentation on genetic basis of anthracnose resistance in tomato. So we all know that resistance, the anthracnose is a major disease in tomato, and you excellently explained the genetic QTL mapping, GWAS, QTL sequencing, PACE, and CRISPR. And these are the, at a molecular level, what are the genes responsible, what are the chromosomes are responsible. Excellent presentation, sir. Thank you very much. Now it's open for discussion, and any scientists, any students can ask, university officers can ask questions. Sir will answer. It is open for discussion.
Sir, good morning, sir.
Hello, Subramanian Garu.
Sir, I'm Subramaniam. So in anthracnose is a major disease in chili, sir. This technique can be utilized in the chili also, no, sir?
Exactly. In fact, our project started with chili, so it's difficult to work with chili, so we worked in tomato. So the PACE assays that we have created can be used for screening chili peppers and identify resistant alleles.
Okay. Thank you.
Yeah, it's very useful that way.
Sir, good morning, sir. Myself Sudhir Kumar Reddy. I'm PhD student.
Hello, Sudhir.
Sir, actually here plants are inoculated with Colletotrichum cuckoides, no, sir.
No, fruits.
So, yes, sir. Actually, these anthracnose causing different species, Lycotruncatum. There are different
Yeah,
species.
that is another huge issue. So we used this Cocodis because this is very important pathogen for peppers and infects tomato. So there are about 25-30 biotypes here also in the US.
Yes, sir. These QTLs are developed now, sir. So these QTLs are resistant against other species also, or only for Cocodis species, sir?
That evaluation we are doing, Sudhir. We don't have data yet.
Okay, sir.
Student is working on it.
Okay, sir. And one more thing, sir. These all developed markers is a flanking markers only, no, sir? Not like a
Markers are
gene
what?
specific marker.
No, gene specific, SNP specific markers. They are located in various genes.
Okay, sir. Mostly these anthracnose related genes are located especially in chromosome 6, sir.
Well, there are all over, but we targeted chromosome 6 because there are more important genes that are having some published background.
Okay, sir.
Sir, good morning, sir. I am Gopal, sir.
Hello, Dr. Gopal Garu. Namaste.
Sir, how is Padma, sir?
Padma is very good and she always talks about you.
Sir,
And then we want to do your memorandum of understanding.
Yes,
We
sir. Yes,
forwarded
sir. It is
to the
waiting, pending
higher
for a long time,
administration.
sir. We want
Yes.
to do it at the earliest.
Absolutely. Yeah. And
Yes,
thank
sir.
you for the opportunity.
Sir. And really, we should thank you because your university and your lab accepted one of our student for PG program, Meghana,
Right.
from College
Yes.
of Horticulture, Anantarajpeta.
Yes.
Really, we are happy for that, sir. And we look
And
for.
also, we want more students from your university.
Yes, sure,
because
sir.
Meghana is doing so well.
We will send,
Yeah.
sir.
So
we
once
want to
we
have
have
a MOU.
students
Yeah.
identified, we can provide them a complete tuition and assistantship so that
Thank
they
you,
don't
sir.
have to spend any money.
Yes, sir. Yes, sir. And now, next year we want to introduce a plant breeding program in horticulture, and
Yeah.
again natural farming, and again pathology, entomology also again restarted this academic year. So we are very eager to have MOU, sir, for benefit of
Yes.
students, staff, and also for having collaborative projects with the.
And also, if
Especially
any
your
students
team
are
with
there,
your
especially for
university.
students, we can give like 3 months or 4 months, 6 months training on our expense.
Yes,
So
sir. Yes, sir.
we
Yes,
can
sir.
sponsor their travel and help them to learn these technologies.
Yes, sir. Even we can have some online courses, lecture also, sir, for
Absolutely.
PhD students, because we have that kind of facility which will be credited for student academic credit bank also. So it will be useful, sir, for both the students and also for the staff.
Yeah,
Sir, here main problems we face in tomato, that's mainly early blight, late blight, leaf curl, and Tospo virus, Badnecrosis virus. These
Right.
four are very major, sir. And again, we want resistant to these diseases with them having qualities required for processing, sir, because we have encountering frequently the glut of tomato production, and that is creating a lot of problem. So we want to have a processing varieties which requires probably the processing units support and also government support to grow for in the farmers use, sir, because whatever the farmers they are using, no, they are not that. You see, earlier old varieties we need to we were having the varieties which have more number of lobes with like a bit sure taste used to be more preferable one among the farmers, among the all the farming community and also consumers. So now processing a little bit processing varieties have come with high yield, but that taste which is probably lacking, and again the resistance for early late blight and leaf curl, the Tospo virus. These 4-5 factors, including shelf life. Yes, sir. So probably we need to have certain projects which can solve these problems, sir, because it is a major things we are facing here. The farmers, especially in the Rayalaseema region, where the tomato is being grown at a large scale in both Kurnool, Chittoor, Anantapur, Kadapa. These areas, it is a major belt for tomato. So probably we need to concentrate probably in that area, sir, in our MOUs. Yes, sir.
Yes, yes. And also we have United States of America International Development. It's an agency, USAID.
Yes,
They
sir.
give funding also for doing some research.
Yes, sir.
like
Yes, yes, sir.
joint
We can,
research.
yes, sir. Yes,
should
sir.
explore,
Here
yeah.
we need your collaboration, sir, to write a collaborative project.
Got to collaborate,
Your
yeah.
university and all, your university and our institute,
Yeah.
so that we can solve these problems, sir, in the coming days.
Yeah.
Yes, sir. Thank you, sir. Thank you very much.
Thank you very much for the opportunity and so many familiar faces. I know Dr. Naram Naidu, I know Shashi Kiran, who was pathologist or virologist around there, and many
Yes,
good
sir.
scientists you have, many good administrators.
Yes, sir.
Thank you very much for the opportunity, and we
Sir.
will be in touch, Dr.
Sir.
Gopal.
Sir, good morning, sir.
Good morning. Yeah.
Myself Seshakiran, sir.
Hello, Sheshakiran, not Sai Kiran, sorry, Sheshakiran. Hello, Sheshu. How are you?
Sir, fine,
Good.
sir.
Good.
Sir, my one small question, sir. Sir, what is the level of incidence of this anthracnose in US, sir? Like out of my
Heavy,
3 years
because this is
work,
one of the most important post-harvesting disease. So a lot of losses happen through this because of anthracnose.
But in coming to our conditions of disease scenario in AP, sir, early blight, septorial leaf spot, these are the major diseases.
That's what
sir, apart from that.
Honorable Vice Chancellor also was saying.
Yes,
A
sir.
lot of issues there.
Okay, sir.
So, Sheshu, we will I mean, contact in emails. Yeah.
Okay, sir. Sir, and above all, this is nowadays, this bacterial diseases, these are all major, sir. I think we can apply whatever these technologies for the tomato crop, sir. Whatever procedures we have outlined. And as Subramanian sir said, this is a major, anthracnose is a major disease in Chile.
Yes.
And tomato,
So
it is
we
not a
also
major
actually
disease.
wanted to work with chili and entered into tomato because it is workable at least. We can do
Okay,
all
sir.
these things. Chili is very difficult to work with. Otherwise, our major work is on chilies.
Okay, sir.
Yeah.
And coming to our tropical
Sir, this is
conditions,
Dr.
leaf curl, this is the major, sir.
Yeah.
Okay, sir. Thank you, sir.
Thank you.
Sir, this is Dr. C. Venkatraman, sir, scientist working in
Hello, sir. Namaste.
at HRS Lam Guntur. Sir, namaste. Sir, is there any line in animal background which is resistant to
Anthracnose?
Capsicum truncatum? Anthracnose, sir. Truncatum for truncatum.
Oh, we are in fact
In
looking,
Anam
in fact,
background, sir.
we want to work with Chinans, which is habanero peppers, because many people are working with
Sir.
Anam in Korea and China. So we thought we will work on the Chinans. So we are using this Cocardis inoculum on Chinans fruits, and then using these markers what we have developed in Chinans. If there is any collaborative.
Cocardis is not a major strain affecting anthracnose in India, sir.
Oh, really? Yeah, that's right.
It's truncatum and gliosporidis, but we are eagerly searching for a stable resistance source for anthracnose for truncatum and gliosporidis.
I see.
We have in buccatum, sir, but it's not compatible with anum.
Yeah, we cannot cross it. We too
Yes,
have
sir.
baccatum. We maintain a lot of pepper germplasm here.
Sir.
There are resistant sources, but the biggest problem in pepper is transferring because of compatibility. So probably we need to use this kind of transgene technology to move the genes or to change the genes.
Yes, sir. Yes, sir.
Yeah.
Sir, good morning, sir. This is Dr. Naram Naidu.
Hello, Dr. Naram Naidu. How are you?
Fine, sir. Fine. Thank you. How are you, sir?
Good.
Sir, just a small query out of curiosity, I'm asking. Sir, in any plant, actually, either it could be a biotic or abiotic stress tolerance is often implicated with the levels of the secondary metabolites and their derivatives. So is the case with anthracnose disease in tomato also. Most of the times, as I have gone through earlier reports when I was doing my PhD, this alpha-tomatin has been implicated, higher levels of alpha-tomatin and its derivatives have been implicated in the resistance of anthracnose.
You are absolutely right, Dr. Naram Naidu. Yes. In fact, a paper is published in Journal of Experimental Botany about this metabolite, tomatacin. And these are the game genes. There are 5 genes. We also have some association.
And gene alterations also to increase the levels of this steroidal glycolides.
Yes. You are right.
My question is, have you attempted any such kind of studies in your research?
Absolutely, we are doing it. We have a lot of metabolite data and about the game genes. All this data we are analyzing and linking with the genomic data.
And my one more question: with all this science and so much of breeding work and resistance breeding and all, why it is becoming very difficult to develop a true tomato commercial variety with 100% resistance to anthracnose? Because few varieties of anthracnose are available in the on commercial scale.
The new technologies have not properly kicked in yet, in my opinion. Very few companies are doing this kind of marker-assisted selection or genomic selection. Most of the breeders are still doing traditional breeding, and genome editing or GMO technologies are still kind of treated with suspicion. So
they're not getting into the
now the government and the policymakers, especially I urge Vice Chancellor, Honorable Vice Chancellor also to convey this to politicians to bring GMOs and genome editing technologies into at least tomatoes, vegetable crops, so that we can reduce the load of pesticides and fungicides on these crops.
Okay, sir. Thank you.
Thank you, Dr. Naram Naidu.
Sir, convey my wishes to Madam Gar also, sir.
Sure, thank you. We will. Yeah.
Right. Thank
you. Sir, one more doubt, sir.
Yes.
Sir, actually in IAVR, one more work I'm doing, sir, related to collar rot in tomato.
Okay.
So still not reported any gene related to resistant against collar rot, sir. Same work is doing in IAVR also.
I see.
So suppose QTL sequence analysis is mandatory to identify resistant genes, sir?
Not like that. The QTL analysis helps you to identify what are the closest DNA markers using which you can select upon. Otherwise,
Okay.
normally also you can screen and identify always resistant sources. That is the first step anyway.
Okay, sir. Actually, in this one, GWAS also we are doing, sir. So we can conclude with that, sir.
Yeah, of course. GWAS and QTL analysis are the baby steps. Then you need to take further.
Okay, sir.
Yeah.
Okay, sir. Thank you, sir.
Good question.
Anybody? Hello?
Hello,
Madam.
sir. Namaste, sir. Sir, is there any released hybrid or variety of tomato in US, sir, which is having anthracnose resistance? No.
We did not do that way in the sense we cannot say this variety is resistant or something. It's all developing pre-breeding lines. In the sense, our job is to develop pre-breeding lines. Say, genome-edited plants are developed, and then the breeding companies and commercial breeders can use them in their breeding program to incorporate the resistance. So it's like that. Several university groups or USDA groups, we develop lines, we characterize lines and genes, and we say these are the pre-breeding lines. And then the commercial breeders use them in their breeding program. So it's a continuous process, madam.
Okay, sir. Okay, sir. Okay. Sir, in generally that Pimpinellifolium, Lycopersicum pimpinelli is having resistance against many number of diseases. Are you using that one only, sir, for as a resistant parent?
This is a great question. In fact, we got intragressid, Pennalia intragressid lines, not Pimpinellifolium, because it is little bit further from the Lycopersicum. So from the Pennalia, we have intragressid lines. The entire Pennalia genome is part by part intragressid in Lycopersicum. So we are growing them in our greenhouse and inoculating those fruits. As you say, one grad student is working on it to identify any new sources in the wild species.
Sir, thank you, sir.
Thank you, ma'am.
Anybody from the audience? If nobody is there, we'll conclude with vote of thanks. Definitely, sir. Thank you very much. And we will have definitely MOU, and our Honorable Vice Chancellor has pointed out that this early blight is a major disease, topsovirus and late blight. We will have a collateral research projects after signing MOU with the West Virginia State University. And definitely in future, we'll enhance our relationship with West Virginia University after MOU. And thank you very much, sir. Excellent presentation. And now I invite our registrar, Dr. B. Srinivasulu, to say formal vote of thanks.
Okay. Thank you, Dr. Dhananjay Rao, Director, Industrial and International Programs, Dr. YSR Horticulture University. Good morning and namaskar to Dr. Umesh Reddy sir.
Namaste.
Good morning, sir. Sir, greetings from Dr. YSR Horticulture University.
Thank you, sir.
Today, this is the 20th Global Connect Series program Dr. YSR Horticulture University is organizing for the past 3 years. And today it is a very informative lecture we have on the topic genetic basis of anthracnose resistance in tomato. Very nice presentation, sir. You have given very clear picture of genomics of tomato towards resistance to anthracnose disease, which is a major post-harvest disease in tomato. I think the work conducted on this QTL mapping, GWAS and CRISPR lines development, and you have given the very clear, the picture of genomics of the resistance towards this anthracnose disease. Very, very useful lecture, both for the faculty and the students. And we, at the outset, I would like to extend our heartfelt thanks to you, sir, for readily accepting our invitation to be the guest speaker for today's 20th Global Connect Series program. We thank you, sir. We immensely thank on behalf of our Honorable Vice Chancellor, Dr. K. Gopal Garu, and all the university officers present over here in this program. And also I express our sincere thanks to you, sir, on behalf of all the faculty and students. I think with your wonderful, the presentation with very clear, pictures of this QTL mapping genome technology and development of CRISPR lines. I think it was very useful lecture today we have. I once again thank you very much, sir, for giving very informative lecture on this genomics of anthracnose resistance in tomato. Thank you, sir. Thank you very much.
Thank you for the opportunity.
Thank you, sir.
Thank you.
sir.
Namaste.
Thank you, sir.
Namaste to all, and wish you all Happy New Year.
Thank you.
Thank
Thank you,
you,
sir.
sir. Thank you, and wish you the same, sir.
Thank you.
Thank you, sir. Sir, Umesh Reddy garu, thank you very much. I think the time is 12:30 night.
Yes,
Sir,
thank you. Yeah.
thank you very much, sir. Thank you once again,
You
and
are
all university officers and all associate deans, all university scientists. Definitely in future, after signing MOU, we will have good relations, research projects from our USDA, and definitely we will go forward, sir. Thank you very much.
Thank you all. Take care.
Now the program is ended.
Thank you, sir.
దాని మీద బేస్. సరే సరే సరే. అవును, మనం ఇది దీంట్లో మళ్ళీ ఇప్పటికే ఎక్కువైపోయినాయి. అది మళ్ళీ ఇంకో దాంట్లో పెట్టాలా. 400 దీంట్లో పెడదాం అనుకున్నాం కదా ట్యూబ్లైట్ది. ఇప్పటికే చాలా వచ్చినాయి. వేరే దాంట్లో దేంట్లో కలుగుదాం.